With that, I'd like to introduce our first speaker. We have Garry Cuneo from Luminex Corporation. And the title of his talk is Luminex Flow Cytometry, Complexity Simplified.
Thank you. Can you hear me all right? I can't see my button.
Yep, you're all good.
Okay, perfect. So thank you everyone for attending. My name is Garry Cuneo and I'm the Northeast Field Application Scientist with Luminex Flow. Today I'm going to be presenting Luminex Flow Cytometry, Complexity Simplified. Our agenda is going to begin with an introduction to our full flow cytometry portfolio. We're going to continue on to Guava Microcapillary flow cytometry, which covers both the Muse, a simple, affordable flow cytometer and a miniature footprint, as well as the Guava EasyCyte, a flexible, intuitive flow cytometer able to meet all your analysis needs. And then in closing, we're going to discuss Amnis imaging flow cytometry and our new intuitive image-focused analysis for the IDEAS 6.3 analysis software. So welcome to our full flow cytometry portfolio. Our flow cytometers and cellular analysis instruments provide instant access to all facets of cellular phenotyping and morphology. Our innovative Guava microcapillary technology in the Guava Muse cell analyzer is able to provide miniaturized systems for quick cell counts, viability, basic cell health analyses, or you can also step up to the Guava EasyCyte system, which is a versatile benchtop platform for additional multidimensional cell health assessments. With the Amnis technology, the cell stream is able to provide unparalleled high sensitivity and a highly customizable flow cytometer, while our FlowCyte and ImageStream imaging flow cytometers add quantitative microscopy imaging to every cell of your flow cytometry needs. So this is the Guava Muse cell analyzer. It's a simple, affordable flow cytometer with a miniaturized footprint. And the Guava Muse features flow cytometry technology and this nice, affordable, compact instrument. Its simple and effortless operation are highlighted through the use of an intuitive cell analysis software coupled with the touchscreen user interface. The Guava Muse enables quantitative data collection at the single cell level and 27 optimized RUO assays, in addition to the new optional open platform modules, the detection of PE and PE-Sci5 in user-defined applications. For the assays you rely on most, we've developed optimized kits, and these include count and viability, cell cycle, a Nexin 5 and dead cell, as well as basic immunophenotyping kits. Our assays are validated for robust performance on the Muse cell analyzer. Typical cell preparation protocols have been condensed and simplified, so your sample preparation will be fast and easy. You don't need to optimize any of the software settings. The Muse instrument calculates all the gating parameters and thresholds for you. You'll spend less time with experimental setup, avoid reagent waste, and save money. The Muse features a highly intuitive touchscreen interface that's going to allow for step-by-step operation. So easy that no flow expertise is required to run these assays. The touchscreen prompts you through simple on-screen instructions and guides you through sample loading to simple setting adjustments to results in just a few steps. The Muse software is designed to be simple enough for any user to follow setup instructions to achieve sample acquisition on the first use. And then assay-specific software modules include sample graphs to make population gating setup foolproof. Results are displayed in both graphical and statistical formats specific to each application, making analysis unambiguous. And then we have easy-to-follow data readouts, and these are going to include summary data and optional dot plots that are specific to each kit that you're able to run. The software allows for PDF readouts, as well as exporting of the FCS file. The Muse count and viability reagent allows for quantitative analysis of cell count and viability on the Guava Muse cell analyzer. It's a rapid and reliable alternative to trypan glue exclusion. This kit is able to provide absolute cell count and viability data on cell suspensions from a variety of cultured mammalian cell lines in a simple one-step no-wash assay. Both viable and non-viable cells are differentially stained based on their permeability to DNA binding reagents. Data generated with the Muse count and viability software provides viable cell count in cells per mil, total cell count in cells per mil, and percentage of viable cells without the need for counting beads. The Muse cell analyzers count cells more accurately than manual hemocytometry or image-based automated analysis. Here we see that cell counts and viability correlate well amongst the multiple methods of analysis. For viability specifically, we compared the Muse to a manual hemocytometer and an image-based cell counter using 5 different cell lines, 2 of which were adherent and 3 are suspension, and then across the 5 concentrations for each. The Muse cell analyzer counts cells and measures viability more precisely with smaller CVs than manual hemocytometry or image-based automated analysis. When compared with image-based automated counting methods and manual hemocytometers, the Muse cell analyzer exhibits a narrower range of percent CVs and consistently provides variation of less than 10% over the entire range of samples tested. Higher percent CVs were observed for the trypan blue-based methods, particularly at the low cell concentrations. Recently, it's been published that T cell counts directly correlate to a patient's outcome as it relates to COVID-19. With the Guava Muse, you can study these lymphocytes quickly with ease in a research use only environment. The Muse human CD8 T cell kit is optimized for use with the Muse system, and it's going to make sophisticated fluorescence-based analysis fast, easy, convenient, and affordable. Sample preparation is minimal using a simplified no-wash protocol, and after loading samples onto the Muse cell analyzer, the intuitive software provides a detailed analysis of your cell sample in just a few short steps. The software readout on this instrument will give you CD8 T cell concentration in cells per microliter, CD8 T cell percent of lymphocytes, and total lymphocyte concentration, once again without the need for counting beads. Here we see an example where human whole blood was stained with the Muse human CD8 T cell kit and acquired on the Guava Muse cell analyzer. Figure A shows a summary of the data, while Figure B shows results displayed with the optional dot plots. The statistics show the percentages and the concentrations for the results in each quadrant out of the total events. The lymphocytes results show the total lymphocyte concentration, CD8 T cell lymphocyte concentration, and the CD8 cell percentage of lymphocytes. The first plot in Figure B displays cell size index versus lymphocytes and a lymphocyte gate, with the second plot showing CD8 versus total lymphocytes. Luminex also offers a full range of Muse-optimized immunophenotyping kits, including CD4, CD20, and more. Now we also allow the open ability to look at increased phenotyping abilities. With the Guava Muse cell analyzer, you can now achieve highly quantitative results at a fraction of the price, effort, and time. The Muse cell analyzer packs 3-parameter analysis into a compact, easy-to-use benchtop device, making flow cytometry accessible to anyone, anytime, with a user-friendly touchscreen interface, intuitive cell analysis software, and optimized assays designed to simplify your research. The Muse is the perfect cell culture and core lab companion. So switching gears a little bit, we're going to move on to the Guava EasySite. Our microcapillary flow cytometry systems are simpler to operate than traditional sheath fluid-based instruments and far easier to maintain. They utilize small sample volumes, generate minimal waste, and have a lower operating cost. As a result, the Guava EasySite flow cytometers are uniquely amenable to on-demand use in the laboratory environment and have helped many scientists achieve insightful cellular analysis since 1998. Microcapillary flow cytometry avoids the complexity of microfluidics seen in traditional sheath fluid-based flow cytometers and allows for a compact, low-cost instrument. The microcapillary format reduces waste and simplifies operation, so the instrument doesn't require a dedicated user. Moreover, it uses low cell numbers, which reduce reagent usage and the amount of cells required, saving critical resources while still incorporating absolute counts. So to demonstrate the capabilities of the EasySite, I have a 8-color immunophenotyping panel to display. And for this particular panel, we used 2 of the total 4 available detectors off the violet laser. We used every detector off the blue laser and both of the detectors off the red to create this panel. 10 microliters of human blood was stained with a cocktail of immunophenotypic markers for 20 minutes at room temperature, and then followed by lysis with Guava lysing solution for 15 minutes. After lysis, the samples were acquired on the Guava EasySight 12HT, which is our 12 fluorophore instrument, using the Insight software. Gating was done with using side scatter versus CD45, and we then gated on the CD45 positive lymphocytes. And then the CD45 positive lymphocytes were then plotted into a side scatter versus CD3 plot. And then we're going to further analyze the CD3 positives and the CD3 negatives. And a CD3 positive gate was used to identify the CD4 positive and CD8 positive T cells. While on the negative CD3 cells, they were then plotted using CD19 to find B cells and CD16 and 56 to find our NK cells. As the figure shows, separation is easily visible and all major populations can clearly be distinguished. The ability of the Guava EasySight 12 to accurately determine subpopulation counts and percentages without the use of external beads was evaluated. Here we observe total lymphocyte concentration, as well as cell concentrations and percentages of lymphocytes for T cells, CD4 T cells, CD8 T cells, B cells, and NK cells for 2 different donors, as well as the standard deviations without the need for counting beads. CD4 and CD8 naive memory cells were further discriminated by evaluating each population in a CD45RA versus CD62L plot. Once again, we show cell concentrations and population percentages for naive and memory subpopulations on CD4 and CD8 T cells without the need for external beads. Microcapillary flow cytometry avoids the complexity of microfluidics seen in traditional sheath-based flow cytometers and enables a very compact, low-cost instrument without sacrificing performance. The microcapillary format reduces waste and simplifies operation. And in summary, the Guava EasySight has the ability to quickly analyze heterogeneous mixtures of cells in 3 to 12 fluorescent channels with absolute counts and optional 96-well plate automation. And now we're going to switch over to our imaging flow cytometers. So these are all of our Amnis imaging flow cytometers. The revolutionary Amnis imaging flow cytometers combine speed, sensitivity, and phenotyping abilities of flow cytometry with the detailed imagery and functional insights of microscopy. This unique combination enables a broad range of applications that would be impossible using either technique alone. Amnis imaging flow cytometers are benchtop multispectral imaging flow cytometers designed for the acquisition of up to 12 channels of cellular imagery. By collecting large numbers of digital images per sample and providing a numerical representation of image-based features, Amnis technology combines the per-cell information content provided by standard microscopy with the statistical significance afforded by large sample sizes common to standard flow cytometry. With these systems, fluorescence intensity measurements are acquired as with a conventional flow cytometer. However, the best applications take advantage of the system's imaging abilities to locate and quantitate the distribution of signals on, in, or between cells. So here's a few of the applications possible with Amnis imaging flow cytometry. Cell signaling is a big one for us, so the NF-kappa B translocation application. Also, internalization in phagocytosis. So with internalization, we can look at the phagocytosis of viral particles, which has been a fairly hot topic these days, bacteria, extracellular vesicles, and other particles. We can do intracellular colocalization, shape change, cell-cell interaction, and a whole bunch of other applications. And then as well, there are some emerging ImageStream applications. Some of these aren't so emerging anymore. They're more high impact. So some of note include the protein aggregation detection for vaccine development. So we can look at protein aggregates. We can also look at protein silicone oil interactions as well in there. as well as microparticle quantification. And coming soon, we're going to have a small particle detection for the image stream that's going to enable enhanced sensitivity. So keep an eye out for that in the future. Morphologically rich image data offers up a wealth of information that you can mine using advanced image processing algorithms in our ideas analysis software. And without going into granular detail, Ideas is able to provide up to 2,000 different features from which to choose from to analyze your data. And these consist of 86 base features per image, and those are size, shape, texture, signal strength, location, and comparison features. And then you can combine those with the masking algorithms, which are going to define pixel intensities over background in the image to be analyzed. And then once you have these, you have the ability to combine masks, combine features, and combine both to make your image analysis very robust. While we have the advantage of the ability to discriminate different cell types based on their imagery, the dilemma for new users is that it's difficult to imagine learning how all of these features and masks work, learning how to apply them to different applications, and choosing the best analysis for an experiment. In an effort to make imaging flow cytometry accessible to new users and to provide high value to power users, we're pleased to introduce machine learning. No longer are researchers burdened with the task of learning how to utilize the 2,000 plus features and masking algorithms to unlock the full power of imaging flow. Machine learning simplifies complex analyses by making it easier to classify cells based on specific image-based parameters in a click, tag, and train model. The machine learning module creates a custom classifier or super feature comprised of many features defined by the user selected images. In turn, this helps researchers customize image-based analysis for their specific application without the need for complex masking or feature generation. So with machine learning, you can simply click on your cells of interest and the software will identify other cells with the same features. So how does this work? Well, the machine learning follows a wizard-based approach for analysis. And for those who are familiar with Ideas, it's similar to the feature finder workflow. It's built on the linear discriminant method, which uses a linear combination of features to characterize or separate two or more classes of objects or events. The resulting combination may be used as a linear classifier or for dimensionality reduction before later classification. The classifier is not a black box, meaning we are going to show you exactly what's being used to calculate your analysis. So if it uses a specific subset of masks and a specific subset of features, we will give you those, and you'll see that as we move on in the presentation. Also, machine learning is fully integrated within Ideas, allowing for these custom feature outputs to be plotted for statistical analysis and saved within templates for batch analysis, and then to be used at later dates. The nice thing is you can constantly train these as well and make them more and more reliable over time. So this is the workflow. And just to basically summarize kind of what's displayed here, machine learning normalizes all the features to a common base to offset the wide variation in the feature value ranges. We then use the Fisher's discriminant ratio for feature selection. Basically, a high RD value is going to indicate better population separation by that feature. And then using a weighted linear combination of the top selected features, the software is going to generate a linear classifier. And you'll see a readout just like you see on this screen. So for this particular one, we're defining the difference between normal cells and blast cells. And then the classifier will be configured such that cells exhibiting characteristics like the positive truth set will score positive, and cells like the negative truth set will score negative. So you'll get a bivariate. So the key steps in this is, first, we're going to perform basic flow cytometry. And we're just going to make sure that we find our focused events, and then we're going to find our single cells or our doublets, and then we're going to do basic immunophenotyping until we're left with the need to find one or more features to define our populations. We then visually tag a minimum of 25 objects for each truth population, and then we're going to select the features to be used. So we have the option to select which size, signal strength, shape, texture, and location features we actually want to consider. So if you're looking for a size change, you can just simply look at size or morphology features. Machine learning then allows the user to take advantage of all the channels or select a select subset of interest, which is going to be 1 to 12 total channels. The module is then going to generate all the masks and features based on your input. Weight the features accordingly and combine them to achieve the most statistically significant Fisher's discriminant ratio. And at the end, you are super happy because you didn't have to do all of the masking and the feature generation manually. So some application examples just to show you how this all works. So one application is sickle cell detection using bright field and side scatter. This is a pretty difficult analysis to perform if you're not familiar with imaging flow. So what we're going to do is we're going to generate a classifier based on our visual cues. So here you can see round cells, sickle cells, and the one other thing that we get with image flow is sideways cells, because all of the red blood cells aren't perfectly aligned to the camera. And we need to separate these so we can be able to determine, okay, what's the rate of sickling. So we load these in the machine learning classifier, the machine learning module. It outputs these classifiers. And what you'll see is you see these bivariate plots. And the plot that you'll see to the the peak that you see to the far right in each plot is your positive situation. So you can see your round, your sickles, and your sideways. We can then plot this, so you now have a sickle classifier and a sideways classifier. And what you can see is that we're able to determine each subpopulation of this heterogeneous main population to pull them out without having to generate all of the massive features to do so. And then just to show that it does work, here are some representative bright field and side scatter images from each of the populations. Another way that you can also use this is to look at mitotic events. So what we did here was we generated classifiers using features from the machine learning module. So to start, we simply use DRAC5, and we're looking for the mitotic events. So we gate on our G2M population. And then once we do that, we can tag some cells, and we tag cells that look like they're going through mitosis and cells that are not. And then we can generate a classifier based on that called the interface classifier. And then to take this one step further, looking at those mitotic events that you see gated in plot B, we can then train the software so that we can visually tag the images that are in metaphase, prophase, as well as anaphase, telophase. And once we have all those truth populations, we load them up into the machine learning module. And here you can see what it will output your populations. So not only will you get all of these images, you will also get the dots to correspond to all of this. So it greatly simplifies your analysis. So the key takeaways from all this is that the machine learning module is based on the linear discriminant method, and you're able to create these custom super features targeted to your input populations or your truth populations. It's going to follow an easy to use wizard-based workflow, and it's fully integrated into our ideas software. And the big positives to this is that it significantly reduces ramp up time for new users and is able to provide fast, high value for power users by increasing their efficiency. And then in closing, just to summarize kind of everything we covered, Luminex offers a full portfolio of cytometers designed to simplify your current research. From the Guava Muse, we're able to provide a simple, affordable miniature flow cytometer that is the perfect core lab companion. We have the Guava EasyCyte, which is a flexible, intuitive, and affordable flow cytometer designed for all your needs. And then we're also simplifying the image cytometry workflow with machine learning by helping you create custom classifiers targeted to user input populations and greatly simplify designing and analysis workflow. We're constantly striving for ways to reduce the complexity of flow cytometry, and keep an eye out for further developments on all of these product lines arriving later this year. So I want to thank everyone. I want to thank the MetroFlow Organizing Committee for allowing us to present during these uncertain times. And if you guys need any more information, please feel free to contact Todd Solomon for our flow cytometry portfolio and Dr. Daniel Morey for our Amnis imaging flow cytometry. And at this point, I'd like to open it up to questions.
Okay, thank you so much, Gary. That was a wonderful talk and right on time. So we have about 5 minutes for questions. And I just want to remind everyone, if you have any questions throughout the duration of the day, please utilize that Q&A function that's right at the bottom. So I'll start off with a question. I'm kind of curious as to what the QC procedure looks like for both the Guava and the imaging unit, so and the Amnis. So what is the QC like for those two?
So the QC is we have easy check or system check beads. So the Muse has system check, the Guava has easy check beads. And what we do with those is they are provided at a known concentration, and they also have MESF values associated with them. So you'll run these beads at the beginning of every run to confirm that you are actually getting the correct concentration of counts, because that's the most important thing when you're not using counting beads alongside your sample, is you need to make sure you're getting these nice robust counts. So that's for both the Guava Muse and the Guava Easy site. We also check the MESF values to make sure the lasers are staying within range. And then on the Amnis side, there's a lot of QC calculations involved because we need to make sure our images are always in focus. So we're running speed beads alongside of the sample at all times to make sure that the core stream is in focus to the objectives. In addition to that, we also calibrate, if you have 3 objectives, we're going to calibrate the 20, the 40, and the 60x objectives. And then we also make sure that they're all aligned to the flow cell itself. So there's a series of about 40 tests on that the Amnis actually goes through on the QC side.
Perfect. Now we have a question from Evan Jellison up in Connecticut, but I guess now Massachusetts. He's asking if the idea is machine learning module, is it cloud-based or is it installed on a local system? And if it's local, are there system requirements or recommendations?
So it is installed locally. We're not planning to farm it out to a cloud. And there's no additional system qualifications that you need to run it. What the machine learning module is actually doing in the background is it's creating all of the masks and all of the corresponding features for you instead of you actually manually creating them and calculating them. So it's no larger a strain on a computer than our standard IPS is.
Okay, great. And then what are some, Tina actually had a question, what are some of the challenges in transferring an experiment from traditional flow cytometry to imaging flow cytometry? And what resources might be available to assist our researchers that are looking to make that transition?
So I think the biggest hurdle that we see from our end is the sample concentration. So the recommended sample concentration on the ImageStream side is 20 million cells per mil, or 1 million cells in 50 microliters. So step 1 is just making sure your sample's concentrated enough. And then it's on the panel development side, because our filter stack is a little bit wider than traditional flow. You also have to make sure you're properly titrating your antibodies, because we are using a camera-based system. The compensation's slightly different, and you just want to make sure that, you know, if you're using, say, FTSE and PE, that your PE isn't, because we control the laser powers and not the detector gains, that your PE isn't so bright that you need to turn down the power on your 488 laser to the point where you don't see your FTSE fluorescence. So titration's also going to play a big role in that. As far as things that you can use to help your user base, the ISAC community has a full portfolio of guidelines for imaging flow. And if you have additional questions, you can feel free to reach out to me at any time, or our support team, and they'd be happy to assist as well. And then in addition to that, we provide with every instrument these experimental design documents, so that it makes it easier for new users to understand how the instrument works and reference planning out their panels.
Wonderful. We have one last question in about 25 seconds. So let's see. We have Jay Enton who's asking what kind of timeline is there for sorting capabilities from imaging instruments, and he thanks you for your talk.
Yeah, thanks for that question. We actually get that question a lot, and currently there are no plans for a sorter in the Luminex portfolio, but we're always assessing the needs of researchers. And if there are any developments, we'll be happy to be on top of those.
Okay, perfect. Thank you so much, Garry. Really appreciate it. That was all the time we had for questions. So thank you so much for a really wonderful presentation.
Thank you, guys.